The cocklebur Xanthium strumarium is a short-day plant that begins to flower in the late summer and fall when day length no longer exceeds 15 hours. In experiments by Carl Hamner and James Bonner, cocklebur plants were grown continuously on a 16-hour light, 8-hour dark cycle. Under these conditions, the plants remained in a vegetative, non-flowering state indefinitely. Reducing the photoperiod by just 1 hour produced plants that flowered abundantly. From these results, it is not possible to determine the critical factor in flowering because the length of darkness and the length of light vary together in this experiment. Hamner and Bonner also experimented with photoperiods that were not on a 24-hour cycle, but extended to 48-hour cycles. In the new treatment, plants received light during the first 16 hours, followed by a 32-hour dark period. Plants flowered under these conditions. Other plants received treatments on 12-hour cycles, with 4 hours of light followed by 8 hours of darkness. Plants did not flower under these conditions. From these experiments, it appears that 9 hours or more of darkness after a light period triggers flowering. Hamner and Bonner's experiments pointed to the length of darkness as important in flowering in cocklebur, with 9 hours as the critical length. To test whether a continuous 9 hours is important, they interrupted the dark period with 1 minute of illumination. This treatment resulted in no flowering. The investigators determined that short-day and long-day plants had a timing mechanism that measures the length of a continuous dark period. Short-day plants require a long night of uninterrupted darkness in order to flower. If the night is short, or if a long night is interrupted by periods of illumination, the plant will not flower. Long-day plants flower when the nights are short or when long nights are interrupted by periods of illumination. They do not flower when nights are long. Both types of plants measure the length of the dark period. The nature of this timing mechanism has been partially revealed and involves a pigment called phytochrome. Phytochrome exists in two forms, a PR form and a PFR form, which refer to red light absorbing and far red light absorbing forms. Red light converts PR, which is inactive, into PFR, the biologically active form. In turn, far red light converts PFR back into PR. PFR also steadily reverts back to the PR form at night. Some of the PFR molecules also decay. Sunlight contains more red than far red light. As night begins, phytochrome is almost all in the PFR form. Over a period of several hours in the dark, these molecules begin to convert back to PR or to break down. Depending on the length of darkness, only some of the PFR remains. If a flash of red light interrupts the long night, the phytochrome molecules reset themselves, converting from PR back into PFR. Night interruptions have opposite effects in short-day and long-day plants. In short-day plants, PFR inhibits flowering. In long-day plants, it promotes flowering. If the night is interrupted by light, with a final flash of red light, the red light essentially resets the night, creating a large pool of PFR forms. This form of phytochrome inhibits flowering in short-day plants, but it promotes flowering in long-day plants. A final flash of far red light essentially eliminates the pool of PFR. Because PFR inhibits flowering in short-day plants grown under these conditions flower. Because PFR promotes flowering in long-day plants grown under these conditions do not flower.